All right, so in chapter 15, we're going to talk about brain and cranial nerves. But the first part of this lecture, we'll start out talking about how the brain develops, as well as the microscopic organization of some tissue in the brain. We'll also talk about how the brain's supported and protected within the cranial cavity. You know, the brain has the consistency of an avocado, and it could potentially crush under its own weight within the cranium, and so it needs to be supported and protected by a series of tissues that we'll talk about. Now, what we'll do after that then is move into each of the brain regions and talk about anatomically what they look like and the functions of each. So we'll talk about the anatomical structures of the cerebrum and the functions of those structures, and then move into the diencephalon, brainstem, cerebellum, the limbic system, and then wrap up with the cranial nerves. There's 12 of these. The reason 12 pairs, the reason why they're called cranial nerves is that they emerge from the brain, hence the name cranial nerve. Okay, and we'll talk about spinal nerves next time, but this week we'll talk about cranial nerves. So we can divide the brain into several key areas. We got the cerebrum, which is sort of the top, superior bulbous part of the brain. We got the diencephalon, which is deep within the cerebrum. We got the brain stem, which is like what it sounds like. It looks like the stem of the brain. And then we have the cerebellum, which they call the hindbrain. It actually occupies about a third of the brain's mass, and you find it just on the posterior side of the brain. That's the cerebellum. And we'll go through the structures and functions of each of these brain regions in the following slides. Now, superficially, when we look at the brain exteriorly, we can see gyri and sulci. The gyri are like the hills on the surface of the brain, and the sulci are the valleys. So when you think of like a gyrus, it's like a hill on the surface of the brain, and the sulcus is the valley. And other anatomical words we use to describe directionally, we use rostral and caudal on the brain. Rostral means towards the nose, and caudal means towards the tail. This is often used interchangeably with anterior versus posterior, although it doesn't really mean the same thing in humans. We'll still say rostral caudal is the same as anterior posterior with respect to brain regions. Now, looking at the surface of the brain here, you guys, what's the hill called again? A gyrus, right? Yeah, gyrus. And then the valley? Sulcus, yeah, exactly. So gyri are the plural, sulci are plural, but it's a gyrus that's singular and sulcus that's singular. So the gyrus is the hill and the sulcus is the valley. You might wonder, why does the brain have this sort of lumpy appearance? Why isn't it smooth? Well, by having more of these folds, it gives the brain more surface area for neurons, and you can actually have more brain cells within this extra surface area locked up in folds on the outside of your brain. In fact, if you have more of these gyri or sulci, then presumably you have more brain matter. And so when they looked at like the brain of like Einstein's brain post-mortem, it turned out he had a lot more gyri than the average person. Like he had way more folds. His brain was highly more folded. So, based on that, we can make the assumption then that he had more surface area on his cortex, so the outer part of his brain, and therefore probably more neurons and more connections, which could have aided in his ability to, be a forward thinker. Now, on the underside of the brain, we can see our cranial nerves, which are in yellow here. Now, they won't really look yellow in dissection. They typically just show nerves as being kind of yellowish, just to kind of make them look more distinct. But for the cranial nerves, we have 12 distinct pairs of nerves that all go out to structures of the head and neck and abdominal cavity, and they're involved with sensation and motor functions. And we'll go through the functions of these nerves later. Now, looking at a mid-sagittal view of the brain, we can see if you cut the brain right in the midline and look at it from the side, you can see the cerebrum, which is the outer part here. We can see our diencephalon, which is in the middle. The diencephalon is divided into our thalamus, our hypothalamus, and epithalamus. We can also see our brainstem really well here. We got our midbrain, our pons, and our medulla oblongata, as well as our cerebellum nearby. But if you guys look closely here, do you guys notice there's some spaces in here? Like right here, there's a triangular space, which leads into a little canal. That leads into another space up in here, and then actually leads into another space that's even deep in the cerebrum. These spaces are called the ventricles of the brain. And it turns out that our brain is not solid. There are large fluid-filled spaces inside the brain that help make the brain more buoyant, less dense, therefore it's able to float within the skull more easily. That way it doesn't crush under its own weight. So we call these the ventricles, and they're filled with not air or blood, but they're filled with cerebrospinal fluid, and we'll talk about what that is here in a little bit. Now, if microscopically, when we look at brain matter, there's really two types of brain matter. We got gray matter and white matter, right? And then, was that Breaking Bad, gray matters? Yeah. Gray matter, yeah. So gray matter is basically the parts of the brain tissue where you have large collections of neuron cell bodies. So a lot of neuron cell bodies that are in a tight area. Because these neuron cell bodies have a lot of protein, it makes the tissue look more gray. Okay, so we call it gray matter. So you got a lot of neuron cell bodies. You're typically going to find gray matter in the cortex, so the outermost layer of the brain, or deeper in nuclei. And nuclei are collections of neuron cell bodies deep within the central nervous system. Now, the white matter appears to be more white because it's made of myelinated axons. Remember, myelin was basically lipids, and lipids are white. It's fat, right? So white matter, because it's mostly myelinated axons, appears to look more white. And so you find white matter typically deeper in the brain, not necessarily on the outer layers, which is the cortex. That's going to be gray matter. But deeper, you're going to find white matter. So what does this look like? Well, out here, it looks darker, so what are we going to call this? Gray matter, right? And what's gray matter made of? Mostly neuron cell bodies. So when you think of the cell body of a neuron, you're going to find that in the gray matter, as well as unmyelinated axons. And then what's white matter made of? Yeah, myelinated axons. And you have these huge bundles of axons, you guys. I mean, there are, billions, hundreds of billions of these axons, or more, actually probably in the order of trillions, of axons that extend all throughout your brain. So it's a really complicated interconnected network of cables here that, once they're myelinated, forms this white matter. And you can find that sort of deeper here. Now, if you see a collection of gray matter deep in the brain, we call this nuclei. So we start talking about the nuclei later. You'll know it's actually a collection of gray matter somewhere deeper in the brain. Okay, so here's a nucleus right here because it's deep in the brain. But this is still considered cortex, so we don't call it a nucleus, although it's gray matter. And you'll notice that there's still gray matter and white matter all throughout the brain. So in this cut, we're looking at a cut through the, kind of a coronal cut through the anterior part of the brain here. Otherwise, we look at different cuts as well, like through the brainstem. We have white matter deeper and gray matter superficially. You can see that over here. And on the spinal cord, it actually flip-flops. So if you look at a cross-section through the spinal cord, you can see gray matter is the deeper type of tissue, and white matter is more superficial. And we'll talk about that in more detail next week. Now, just some different terms before we move forward, you guys. A ganglion is a collection of neuron cell bodies outside of the central nervous system. So what was the central nervous system? Brain and spinal cord, right? And so if it's anything else other than the brain and spinal cord, then it's the peripheral nervous system. I mean, anything else that's made of neural tissue, right? So if a collection of neuron cell bodies that are not part of the brain and spinal cord, we call that a ganglion. Okay, so we'll talk about cranial ganglia and spinal ganglia later. But ganglia are collections of neuron cell bodies in the peripheral nervous system. There's ganglia all over. They're like tiny little brains that you find throughout your body. Now, a center is basically just a group of neurons with a common function. So we talk about brain centers. So if there's like a brain center for vision, that's just a group of neurons that have a common function. That's what we call centers. A nucleus is just a collection of neuron cell bodies, deep within the brain itself. A nerve, we talked about last week, it's basically a bundle of axons in the peripheral nervous system. And a nerve plexus is basically an interconnected network of nerves somewhere in the peripheral nervous system. A tract is a bundle of axons in the central nervous system. So we'll talk about the different types of tracts later. Like there's tracts that go side to side, up and down, or just locally. And these tracts are involved with basically communication, around your brain. We have funiculi, which are basically tracts that you find in the spinal cord. There's pathways, which are basically centers and their associated tracts. And the cortex is the outermost layer of the brain. So remember, cortex means bark, like bark on a tree. So the cortex of the brain is the outermost layer of the brain. And that's what's going to basically have your gray matter. And that's going to have their neuron cell bodies. Now, the cerebral hemispheres are essentially the two halves of your cerebrum. which we'll go through in a little bit. So what we're going to talk about first, though, you guys, is basically how the brain is supported and protected. We know the brain has the consistency of, living brain tissue has the consistency of avocado. And knowing how soft avocados are, they're pretty easy to bruise. If you go to the grocery store, sometimes you can find avocados where people squeeze a little too hard. You're like, oh, dang it. You can just tell, like, there's like a little thumbprint area where someone squeezed a little too hard. There's like a soft spot there. Well, it's a really soft tissue, and it is. And so knowing that the brain is so soft, it could be potentially damaged if it moves around too much within its cranium. It needs to be supported and protected in a variety of ways. It's not sufficient to say that the cranium can adequately support and protect the brain. Sure, yeah, our bony cranium does surround our brain and protects it, but to an extent, because the bony cranium itself is hard, right? That's like saying, oh, my avocado is safe here within, I don't know, within like my lunchbox, right? My avocado is safe in my lunchbox because the lunchbox is like a hard outer layer, and you got the soft avocado in there. But what if you start shaking that lunchbox around, and the avocado starts bouncing around the inside? Well, the avocado can get bruised. So it's not sufficient to say that your brain is totally protected by the cranium, because the cranium itself could potentially damage the brain. In fact, the brain weighs several pounds, and if it couldn't float inside of the skull, it would start to crush under its own weight. In fact, when we go to the coroner later, and we actually have a chance to hold fresh brain tissue, you'll see that as you're holding it in your hands, it will start to mold between like your fingers, and it starts to kind of sag down on itself because it's not even floating anymore. In fact, when we look at this model here, and it all looks like a nice kind of stiff position, if this were fresh brain tissue, it wouldn't look like this. It would look really saggy. That's how soft it is. Now, we'll have some experience with working with brain tissue next week with sheep brain dissection, but those are fixed in formaldehyde, so they're going to be a little bit stronger and tougher than like living brain tissue, but you can still kind of see how weak it is. It's got the consistency of like a soft cheese. And yes, the brain is protected by a bony cranium. I know, yeah.
I feel like there's no other comparison you can make. But
there's not.
Even to food, it's like, oh, great.
Exactly.
Oh, great.
Just don't think about it while you're eating. Now, we can't say that the cranium is it. So there's got to be more to it. So the brain is actually protected by a series of tissues called the meninges. So the meninges are a series of connective tissues that surround the brain, and they basically help anchor the brain to the skull, but they help keep the brain in position. So if you all have heard of like meningitis, well, that's inflammation of the meninges. Well, meninges are tissues that surround the brain. So if you open up someone's skull, you're not just going to see the brain right away. You'll see the meninges, and the meninges surround the outside of the brain. You need to remove those meninges before you even get to the brain tissue. Okay? Now, what also supports the brain is cerebrospinal fluid. We talked about last week how ependymal cells, which are a type of glial cell, they take blood. They can basically modify blood plasma to make cerebrospinal fluid, which is essentially like Gatorade. Remember, water, sugar, electrolytes? And so that's what cerebrospinal fluid is, and it helps the brain float within the skull. So if you didn't have cerebrospinal fluid, the brain couldn't float very well. Okay. Now, the brain's also protected by a blood-brain barrier. We talked about this a little bit last week, how astrocytes surround blood vessels in your brain, and they prevent substances in your bloodstream from making it into your brain tissue because there are things in your blood that could potentially damage your brain. You don't want them getting into the tissue. So this blood-brain barrier is also another protective barrier. We'll go into more detail on each of these aspects here. Yes, you have a question?
With the spinal fluid, when you have like a spinal tap, you can get a spinal headache, which I had, and it was like a horrific headache, and they did a blood patch. Is that why you get the headache? Because there's not enough of the fluid around to like keep it where it should be?
Yeah, so the question was, if you lose some cerebrospinal fluid, can that cause a headache? And the answer is yes. If you get a spinal tap and they remove some of that cerebrospinal fluid, your brain stops being able to float within the cranium as well. It starts to pull on the meninges, and that ends up being painful, and you get a headache from that. That's also why you get a hangover headache. If you drink a lot and you get so dehydrated that you can't make enough cerebrospinal fluid to float your brain within the skull, and that gives you a headache. I'll ruin alcohol for you too. Now so if you have a headache after a bender, now you know why. You know, your brain's not able to float. So the meninges, you guys, there's three main layers. So they're connective tissues. That the three main layers. We got the pia mater, arachnoid mater, and dura mater. Their functions are to basically enclose and protect blood vessels that surround the brain, and they help contain and circulate cerebrospinal fluid. They form some of the veins that drain blood from your brain. Like some of the veins that drain blood away from your brain are actually formed in the meninges itself. And they also help separate soft tissue from the bones of your skull, right? You don't want your brain right up against your bone, right? Because if you moved your head at all, then your brain would smash up against your skull, and that could cause a contusion or basically a bruise. So we have three main layers of the meninges: pia, arachnoid, and dura mater. Mater means mother. Okay. Pia means like soft or pliable. So pia mater is a soft mother. So it's the softest, most pliable of these meninges. And you find the pia mater directly on the brain tissue itself. Think of the pia mater as like this external kind of nourishing blanket for the cortex of your brain tissue. Like on the outside of your brain, you get this nice little layer of pia mater that directly surrounds the outside of the brain. Just outside of that, we got the arachnoid mater. When you guys think of arachnoid, what comes to mind? Spiders, yeah. So literally arachnoid mater means spider mother because it has a spider web looking appearance to the tissue. And dura means tough, like durable, right? So dura mater means like the tough mother. So it's the most durable, it's the toughest of these tissues. So I don't know what kind of mother you have, but I guess you can pick from these three. You have a soft mother, a spider mother, or a tough mother. I don't know what that means, you guys. I'm just delirious. So let's look at these tissues in cross section. So what this slide is showing you guys is as if you could cut through the skull and then basically look at what you cut at in cross section. All right. Well, we got our skin of the scalp. Right? Then we'd have some subcutaneous tissue, a little bit. Maybe a little bit of gallia aponeurosis up in there that would connect the epicranius muscles together, right? And then right on the cranial bone itself, we'd have periosteum. That makes sense because all bone is surrounded by periosteum. Then you have your bone. You'd have some compact bone and spongy bone. And then inside the spongy bone, you'd find marrow, whether red or yellow marrow. If it's cranial bones, red marrow. But that's not a good place to get marrow if you're doing a transplant because it's right by your brain. So there's better places. And then let's look deep to that, you guys. Okay, well, if you just go deep to the cranial bone, whoa, okay, now we're in the cranium, right? The cranial cavity. So that's what houses your brain. So the analog to periosteum on the inside of the cranium, we actually call dura mater. So dura mater is the periosteum inside your skull. So we said that periosteum was the tough outer layer of bony tissue. Back when we talked about bone. But periosteum is actually going to be found inside the skull as well, and that's what makes your dura mater. That's why it's tough. If y'all remember, periosteum was a dense, irregular type of connective tissue, and it's a very tough tissue too. So there's two layers of dura mater. We got the periosteal layer and the meningeal layer, and those two layers can separate sometimes. Not all the time, but sometimes. The periosteal layer is what's right up against the bone. That's what we call the periosteal layer of the meninges or dura. Then we have the meningeal layer here that's not next to the bone. It's kind of deeper. Now, normally they're fused together. But in some areas of the brain, these periosteal and meningeal layers can separate, and they form a blood-filled space, which is kind of weird. So between the periosteal and meningeal layers of dura, you can sometimes find a blood-filled space, and we call this a dural venous sinus. And the dural venous sinus is essentially a venous blood-filled space where a majority of blood from your brain drains into. So the jugular veins that drain blood away from your skull basically drain blood. They get their blood from this dural venous sinus. And we'll talk about this more so in lab next week. Now, just deep to the dura mater, we got the arachnoid mater. You can see why it's called arachnoid mater because it's got kind of the spider webs, right? And then just deep to that, we got the pia mater, which is this pink layer. And the reason why it's pink, you guys, is that pia mater, well, it's soft, right? That's not why it's pink, though. It's pink because it's full of blood vessels, right? So the pia mater is basically a big old blanket of blood vessels that surrounds the outside of the brain, and it nourishes the cortex of your brain. So it basically helps bring in nutrients and supplies the outside of the brain with nutrients.
Is it like highly vascular?
It is highly vascular. Well, it's all blood vessels, so yeah, absolutely. So what are those three layers, you guys? What's the outside tough layer here? Dura mater, and then we got our arachnoid mater and our pia mater. Good. Now there's a little bit more to it though. We can add on to that, right? So there's two layers of dura. What's the layer of dura that's right against the bone? Periosteal layer of dura. And then what about this layer? Meningeal layer. And when they separate, it forms a
Dural venous sinus.
yeah, good. It forms a dural venous sinus, which is a blood-filled space you find in the brain. Now, then we have our arachnoid mater here and our pia mater. But you guys notice how there's actually some spaces between these layers. There's actually a space between the dura and the arachnoid here. Do y'all see that? Kind of this little darker area. This is called the subdural space. So the subdural space is a space just between the dura and arachnoid, and it's a potential space, which means the space doesn't normally exist, but it has the potential to fill in with blood or air or something. And that's what potential spaces are in the body. It's like normally the space doesn't really exist there, but it can exist if things can fill it in, right? So the subdural space between the dura mater and arachnoid mater is a potential space, and that can fill in with blood if you have a subdural hematoma, which you guys may have heard of before. So that's where that would exist. And the arachnoid mater is yes, right here, the pinkish line. Yep. And then do you guys notice that between the arachnoid and pia mater is another space here? This space is called the subarachnoid space. So between arachnoid and pia mater, we have the subarachnoid space. This is a true space though, and it's filled with cerebrospinal fluid. So what's the function of cerebrospinal fluid? Nourish, protect in the sense that it allows for buoyancy, and a fluid cushion, right? So when you move your head around, your brain's not just smashing up against your skull, it's actually protected by a fluid cushion between your skull and the brain tissue. So that part of that fluid cushion then is actually the fluid that fills your subarachnoid space right here, which you find between the arachnoid mater and pia mater. And the pia mater is what just kind of tightly hugs the outside of the brain. We will see these in our sheep brain dissection next week. Okay. So let's go through those layers one more time, you guys. So what layer is this? Periosteal layer of dura? Meningeal layer of dura. What space is here? Subdural space. Good. And then we have our arachnoid mater. What space? Subarachnoid space filled with cerebrospinal fluid, right? And then our pia mater, right? What about when the periosteal meningeal layers of dura separate? They form a dural venous sinus where the veins drain into. And so if you think about like jugular veins in your neck, they're draining blood away from that dural venous sinus. But do you guys notice how there's like little extensions of arachnoid mater that go up into that dural venous sinus right here? Yeah. These are called arachnoid granulations or arachnoid villi. And basically this is the place where cerebrospinal fluid gets reabsorbed. So remember, cerebrospinal fluid is found in the subarachnoid space. But if you look here, there's little extensions of that go up into the dural venous sinus. And they form these little clover-shaped structures, or foot-shaped, called them pedunculated. Or you can say they're kind of granular too. I would say they kind of look like little cauliflower structures. If you see them in real life, they look like little tiny cauliflower. Now, this is actually where cerebrospinal fluid gets forced back into the bloodstream. So due to fluid pressure, CSF can get forced from the subarachnoid space back into your bloodstream. That's how it gets recycled. So it's made deep in your brain tissue, but it gets forced back in the bloodstream at the dural venous sinus through these arachnoid granulations. Does that make sense? All right, cool. So you guys want to know the order of these layers. Like if I gave you guys a sequence of these layers, you should be able to put them in their correct order. Okay. And there's actually one more space I forgot to mention. Between the dura and bone, we have the epidural space. You ever heard of an epidural? Right? Not epidural hematoma, those exist. But like if you get an epidural, what they're doing is they're basically injecting, some sort of drug into this space between the bone and the dura. That's the epidural space. Now, if it's around the spinal cord, the epidural space is actually a fat-filled space. Around the brain, the epidural space is a potential space between the periosteal layer of dura and your cranial bone. But it has the potential to fill with blood. So you guys heard of like an epidural hematoma. It exists in that space. It's kind of interesting.
A lot of times, it comes from brain injury, right?
Absolutely.
Yeah.
So what we see here, you guys, is kind of an up-close view of, or actually a better view of all the dural septa. Now the septa are basically extensions of dura mater that go deep into the brain. So you can see all these dural septa. And you guys see all this blue stuff? All of these blue venous-looking structures is actually dural venous sinus. So we have different ones like superior sagittal sinus, inferior sagittal sinus. Those meet up at a confluence of sinuses, which eventually drain into the jugular veins through this occipital sinus. Basically, the dural venous sinus is to basically help drain venous blood away from the brain, but also help reabsorb cerebrospinal fluid. And then the septa, or basically the meninges here that surround the brain, are there to help support the brain itself. And we'll go into more detail on the anatomy of this more so next week. Now, if we look deep in the brain, we actually have ventricles. And the ventricles are fluid-filled cavities deep within the brain. Remember, your brain's not solid. It's fluid-filled. And so we have lateral ventricles that are separated by a septum pellucidum. We have a third ventricle and a fourth ventricle. So if you look at this picture here, what this picture is showing is if you could see deep into the brain, this is what the ventricles would look like. Normally you can't just see through the brain to see these things. You have to chop it up into little pieces and then eventually kind of see that there's spaces there. But if you can reconstruct all those spaces, this is what they would look like deep within the brain. So these big old ones in the cerebrum out here, those are called the lateral horns. The lateral horns you find in each cerebral hemisphere. So if here's one cerebral hemisphere, here's another cerebral hemisphere, there's a lateral horn found in each. Okay, and these lateral horns or lateral ventricles are basically these large fluid-filled spaces in the cerebrum. Now the lateral ventricles connect to the third ventricle, which is what is found between the diencephalon, deep in the cerebrum. And what connects the lateral ventricles to the third ventricle is the interventricular foramen, or foramen of Monro. Okay, so all these fluid-filled spaces are interconnected. And then the third ventricle is connected to the fourth ventricle, which you find in the brainstem. The third and fourth ventricle are connected through what's called the cerebral aqueduct. You can see those structures here. Now, looking at a side view here, lateral aspect, you see our lateral ventricle, third ventricle, fourth ventricle, right? So what connects the lateral ventricle to the third? Interventricular foramen. You might wonder, how do we just go from lateral to third? Well, it's like 1, 2, 3, right? Although these aren't actually numbered 1 and 2. We just call them both lateral ventricles, and the third just happens to be this one. Okay, but you're right. It's the interventricular foramen, or foramen of Monro, that connects the lateral ventricle to the third ventricle. Third ventricle you find deep in the brain. It's a very narrow space. And then what connects the third ventricle to the fourth? Yeah, cerebral aqueduct, right here. And the cerebral aqueduct you find through the midbrain, and it goes through the midbrain of the brainstem down to the fourth ventricle, which you find between the pons and the cerebellum. And the fourth ventricle also links up with the central canal. So all through the spinal cord that goes down your spinal column, you actually find a fluid-filled space in there called central canal. Now, do you guys think this fluid's trapped in the brain? No, it probably leaks out, right? Where else do you find cerebrospinal fluid? Around the brain in the subarachnoid space, right? So it turns out that cerebrospinal fluid's made in the ventricles. It somehow leaves, I'll talk about that here in a minute, and it gets into the meninges around the brain, and it enters a space called the subarachnoid space. Okay. Now, where it leaves is actually at the level of the fourth ventricle. If you guys look here, we have these apertures. We have the lateral aperture and median aperture. The aperture is just basically an opening. It's basically a hole in your brain. It's a space for cerebrospinal fluid to leak out into the subarachnoid space. That way the fluid doesn't just build up inside your brain tissue. Okay? So cerebrospinal fluid is basically a clear, colorless liquid that circulates in the ventricles. It functions in buoyancy, protection, and environmental stability. And it's basically made by choroid plexus. So you find choroid plexus, which is the place that makes cerebrospinal fluid. You find that in every single ventricle. So the lateral ventricle's got them, the third ventricle has it, the fourth ventricle has it. It all has choroid plexus. And basically what choroid plexus is so plexus means like a network. And choroid refers to the blood vessels. So a choroid plexus is a big old network of blood vessels that are surrounded by ependymal cells. And you'll remember from last week, ependymal cells, their function as a glial cell was to basically take nutrients out of blood and turn that into cerebrospinal fluid, right? So it actually takes blood plasma, and that takes the components from that plasma to make CSF. So in cerebrospinal fluid, you find a lot of water, you find a lot of sugar, and a lot of electrolytes. But what you don't have are blood proteins and blood cells. So you won't find red blood cells, white blood cells, or blood proteins inside of cerebrospinal fluid. But you will find a lot of water, sugar, and electrolytes. Think Gatorade, right? Or Powerade, whatever tickles your fancy. So where do you find choroid plexus? All the ventricles. Good. And what are the glial cells that make choroid plexus? Ependymal cells. Good. What's cerebrospinal fluid made from? Blood. And what's it made of? Water, sugar, and electrolytes. Very good, you guys. All right, cool. So what this slide shows is basically the flow of cerebrospinal fluid around the brain. And if you guys look closely, it shows all the ventricles. So we have lateral, third, fourth. And it's showing that there's a directionality of these arrows. If you guys notice that the direction actually goes from lateral ventricle to third, from third to fourth, from fourth into the subarachnoid space. You might wonder, why is there any kind of direction to these arrows? Well, if we go back here, do y'all notice how these ependymal cells are ciliated? Those cilia actually move, they wave, and they create currents of that cerebrospinal fluid within the brain. Those currents help move the cerebrospinal fluid in a particular direction. That's what allows the CSF to flow from lateral to third to fourth, and then into the subarachnoid space where it leaves through the apertures, right? Now, once in the subarachnoid space, it'll bathe the outside of the brain. And then where's the place where CSF gets reabsorbed? Yeah, dural venous sinus, right? But specifically, that little place where subarachnoid space protrudes up into this dural venous sinus, we call that the arachnoid granulation, or leptomeninges. That's where CSF gets reabsorbed. So fluid gets forced back into bloodstream. So where's CSF made? Choroid plexus, which you find in all your ventricles. Where's it reabsorbed? Arachnoid granulations, right? So that's the general tendencies. It's made in the choroid plexus, and then eventually it finds its way to the arachnoid granulations and the meninges. But if it gets reabsorbed through the meninges, what if you have meningitis? Yeah, then you might have difficulty with reabsorption. But you're still making it. There's no communication between where the CSF gets reabsorbed and where it's made. There's no crosstalk there. They're not saying, hey, you're not making it fast enough, make more. Right? Oh, if you're making it too fast, stop making it. Like, it's just going to keep making cerebrospinal fluid, even if those leptomeninges or arachnoid granulations get blocked. And that can happen with infection or trauma, and that can lead to an excessive amount of cerebrospinal fluid within the brain. What do you guys think would happen if too much CSF started accumulating inside your brain? Yeah, pressure. You got it. Like fluid pressure would start to accumulate within your brain tissue. You'd have a rise in intracranial pressure, which could actually be fatal. We call it hydrocephalus, which is kind of interesting.